Today, I will speak about two perinatal brain damage. The first one is prematurity, and the second one, birth asphyxia. But before going into this topic, I just want to briefly discuss with you the importance of the multidisciplinary in these two projects, because as Michael told you, I came from psychology, and today I will speak more about the behavioral and also neuropsychological data. But in these two projects, we also have clinical and some brain and some neuroimaging data. We work with some people from the CIBM, and because we have brain imaging and also EEG. And I think this is thanks to this great collaboration that we could really build some comprehensive projects like the two I will present you today. The first one is called NeoBrain, and it is about the long-term impact of early nutritional and pain management in very preterm infant and then brain and health function. Very preterm means that they were born before 32 weeks. And it was a longitudinal study that has followed 51 very preterm from their birth until they were 8. And we are very lucky because most of them has continued till the end. And today I represent you some of the behavioral data from the 41 remaining kids. The first study is about executive function, so the higher order cognitive process that allow us to generate self-serving and goal-directed behavior. And we know that in the last decade, their survival rate has increased, but they are still at risk to develop a long-term neurodevelopmental impairment in the executive function. They have lower inhibition, shifting, and also working memory skills. In the clinic, we do have some comprehensive battery that assess this executive function, but they last many times. They last hour, and we need to have different batteries that each assess different executive function. But there is an experimental task. This is the flanker task that maybe some of you may know, and it has the advantage to last less than 10 minutes. While giving multiple executive function measures. And so I think it could really be great if we can use it in the clinic. The flanker task is a selective attention task. As you can see here, we have used a child version of it with fishes, and the child has to feed the target fish by pressing a key on the same side from which the fish was looking. We have 3 blocks. In the first one, it was the blue fishes, and the target fish is the middle one. Then in the second block, it was the pink fishes, and the target or the fishes on the side. And finally, with the last block, we mixed both fishes and both rows. And that gave us multiple measures that as the response time and accuracy. The inhibition is the accuracy of the second block because they have to inhibit the previous row, and so their automatic response. The executive function is the accuracy of the last block because we have the two rules. And finally, with the switch cost, we measure their flexibility because it is the ability to switch between two tasks, and it is measured at the difference between incongruent and congruent response time. We also use some standardized clinical battery. The first one is the WISC. It's an IQ test, but we only kept the working memory and processing speed index. I will give you some example. Here it's a picture span, so it's a memory of objects. Then with the digispan, they have to remember a sequence of number, first in the same order, then in the reverse one, and finally in an ascending order. For the processing speed, we have the code. So they have a digit symbol code. They have to then use with a specific key to draw this correct symbol in the correct case. And finally, with a symbol search, they have a line with multiple symbols, and they have to find if the target are present or not. We also use a questionnaire that we gave to the parent, assessing their child behavior. It's about different executive function, and we kept the inhibition, flexibility, and working memory. We use a NEPSI. It's a battery that assesses different skills in the 6 major neuropsychological domain, such as executive function, attention, language, but also the social ability. But we only kept the design fluency because it measures at the same time the cognitive flexibility, working memory, and initiation capacity. You can see here what it looked like. So the child was asked to draw as many unique designs as possible by connecting the 5 dots. And finally, we use a computer-based test. It's a corner. It measures attention capacity because it is a go-no-go task. So the child has to press a key whenever they saw a letter, except when it is an X. And it gives us different measures such as omission, commission, and perception error, but also information about their response time and their vigilance. The first thing that we do, we looked at correlation between the flanker and the clinical measure. You can see here the positive correlation or in blue and the negative in red. And we found nice correlation between the inhibition and accuracy and the working memory and processing speed of the whisk. It was a positive one, and it makes sense as the inhibition and the working memory are deeper and share some common neural pathway. And also, if we can well inhibit, we also will be faster. And we found a correlation with the CPT3. We found negative correlation between the inhibition and accuracy and the perseveration score. And again, if we can't inhibit the previous rule, we will continue to follow them and continue doing the same mistake again and again. Then we compare the results with the result of term children. And what we found that first, even if they were faster, the difference was not significant, but they had lower accuracy and lower executive function score. And that makes sense because it's corroborate past result in that field. And finally, we just wanted to draw the cognitive profile with the clinical batteries. And what we found is that they were in the norm in all of the texts, except for the CPT3, where they are slower and make more omission error. And I think that could indicate inattentiveness, which is a feature related to prematurity. We have a faster growth than we can find in the literature. It is maybe due because none of them was born before 28 weeks. And also when they were neonate, they only have few brain lesions compared to what we can find in past research. The take-home message here is that they have minor difficulty in their executive function, and that the flanker task has a good sensitivity in capturing the executive function and also has a great discrimination of mild difficulty. Because even in a population who is doing quite well, it's able to find really mild difference. And that might allow us to use it in the clinic, also to use it as a screening test because it is fast before going into more investigation. And I think it's really great to have a large variety of tests that we can use in the clinic to be more suitable for each child. The next study, we wanted to see the multisensory processes of very preterm children. It's more about the manner in which the different senses is combined, because being born prematurely and in particular being in the NICU can impact the multisensory processes. It is due to the atypical sound due to the machine, also the lights are brighter, and they have to undergo a lot of painful procedure. And we know that they have impaired multisensory in early childhood, but the question now is if it persists into later childhood. So we wanted to evaluate the integrity of low-level multisensory processes in this population. And for that, we use a simple detection task with visual, audio, and audio-visual stimuli. So the child either saw a cloud or a star and heard a yawn or a yell. And the task, they have to press the spacebar as quickly as possible each time they saw or heard something. And that gives us the response time, but also the multisensory gain. The multisensory gain, it's the audiovisual facilitation. You can see here on the graph. It is the difference between the fast modality response time and the response time in the multisensory presentation. We have compared the data with full-term children cohort, and what we found that they have the same multisensory gain. First, they share their fastest modality was for both visual, and they both benefited from having a multisensory presentation, and their multisensory gain was comparable. But in every condition, they were slower. That could indicate also inattentiveness, but poor processing speed skills, and they are slower. But then we wanted to go a bit deeper and to investigate the underlying multisensory processes. And for that, we use the race model inequality from Miller. So the fact is that when we have two stimuli at the same time, we will have a greater likelihood of getting an answer faster. It's called probability summation. I will give you this example. If someone is rolling one die, the probability of rolling a 5 is 1 out of 6. But if he's now rolling 2 dice at the same time and that the die are independent, the probability of rolling a 5 now is 1 out of 6 plus 1 out of 6, so 1 out of 3. And in our case, it means that the sensory processes is independent, so the behavior will be the probability of a fast response in one modality, visual, plus the probability of a fast response in the second modality, audio. This is a race model, so the stimuli are processed on two different channels, but if there is a neural interaction behind, the probability summation won't be enough. That's what we tested. If the information delivered to the two senses interacts before initiating the motor response. And we do that with the race model inequality formula you can see here. Basically, we compare the observed multisensory data with the data predicted by the model. And if the observed data are lower, that means that the race model is satisfied and that it is independent. But if the observed data are higher, it means that the race model is piloted and then we have this neural interaction. What we found is that in very preterm children, you can see here on red that the multisensory data are lower from the predicted one. But in term children, for the first 3 tentacles, so it means that they are faster, the observed multisensory data will be higher than the predicted one. And that means that in term children, there is an evidence for a co-activation. But that doesn't necessarily mean that there is no co-activation at all in very preterm, especially because, as I told you, they are slower. And we know that slower response time do not activate the co-activation process. And so maybe the fast and the slow response time doesn't show the same anatomic path. And so it is the first evidence that even if they are slower, they share the same multisensory gain, they are slower, and also the underlying multisensory processes are different. And I think it would be really great to make deeper investigation in that field, especially in the underlying neuromechanism by using some neuroimaging, because it has been done with neonate, but not in children. And for the last part, I just want to present the next study. It's a NeuroNeo about perinatal asphyxia in neonates and in children. As Michael told you, we just started this year, so I don't have any result to present, but I will just present what we are doing now. So perinatal asphyxia is caused by cerebral hypoperfusion and a lack of oxygen, and it could lead to neurological insult. It is really important because it is a major cause of neonatal death, and also there is multiple adverse long-term neurodevelopmental impairment. And for the moment, the only neuroprotective treatment we have is the therapeutic hypothermia, but it's not suitable for each neonate. And we wanted to investigate their brain integrity and function using a multimodal neuroassessment with brain MRI, EEG, neurological and neuropsychological assessment, as well as echocardiography. That will allow us to have a better prognostication, but also to individualize follow-up and care. For the neonatal part, we'll compare the brain structure of neonate with encephalopathy and with normal term control. Based on past year, we will expect to have 40 neonates with asphyxia and 30 controls. And for the moment, we already have 6 neonates with hypothermia and 1 without hypothermia. And we will start soon the recruitment of controls. We have a lot of different measures. We will do a routine neuromonitoring with a health ultrasound and EEG to measure the pattern and seizure. They also have a daily clinical neuroexamination and a bigger one before the discharge, assessing muscle tone, reflex, and behavior. They have an echocardiography. We also use a NIRS. And finally, just before the discharge, we make a video to evaluate their motricity. For the neuroimaging parts, they will do a brain MRI after the hypothermia. I won't go into many details because we are still at the acquisition stage and we will work with some collaborator for the analysis. But you have to know that we are looking for 2 types of injury patterns. The first one is an abnormal signal intensity, especially in the basal ganglia and thalamus. And the second one is a watershed pattern of injury. It is in the intervascular boundary zone, mostly in the white matter. But if it is more severe, it will also impact the gray matter. And finally, with the spectroscopy, we will look at the ratio between lactate and acetyl acid. And we will also use the EEG to measure the auditory processes. Neonate will passively hear different sounds, either living, so human or animal sound, and man-made sound, either action or action-free. And I will conclude my presentation with the school-age population. It's about children at 6 years of age who were admitted with a neonatal encephalopathy. We want to assess their detailed neuropsychological phenotype according to their initial brain lesion. Again, we expect to have 40 children, and we already have 6 and 5 are planned on the next month. First, we have to retrieve the neonatal data, especially the MRI1. Then we will give some questionnaire to the parent, assessing the emotional and conduct problem, the executive function, also the sensory processing of the child, but also their own mental health, because we know that impacts the child development. Regarding the neuropsychological exam, we have a lot of different tests. And we will particularly look at the memory because we know that asphyxia impairs it. The first one is a KBC. We can see some examples. So on the upper right, they have to find the intruder. Here it's a memory test. Each sea animal has a name and they have to remember it. Here you can see it's logical sequences. And finally, there is a visual spatial task when they have to recreate the image with blue and yellow triangle. We will use an EPSI for the narrative memory. We will read them a short story. They have to remember, and then we ask some questions. For the memory of faces, they saw a lot of faces, then they saw 3 faces, and they have to pick the one they have already seen. Then for the ray figure, they first have to copy it, then we hide it, and they have to draw it from memory. The CAP-CPT3 is a version of the CPT3, but for younger kids. They have to press the bar each time they saw an object, except when it is the ball. We will again use the flanker and the simple detection task as presented before. And we will monitor the motor performance, and also they will have a neurological examination done by a pediatrician. And finally, they also have a brain MRI. We will look at the amperage, diffusion, resting state, and spectroscopy, but they don't do any task in the scanner because they have a lot of tasks to do except that. And finally, we have 2 EEG tests. The first one is the auditory, the same as a neonate, but here they have to discriminate sounds, so tell us if it is man-made or living sound. And we looked at the ERP component, especially the difference between living and man-made, also between conspecific and other animal. And finally, the difference between action-related and action-free, it's based on past study in that field. And the last task is a zoo task. It's a go, no-go, and error processing task. The child is asked to help the zookeeper to catch the animal that has escaped from the cage, with the exception of the orangutan, who are also helping the zookeeper. And so they have to press the spacebar each time they saw an animal, for the exception of the orangutan. That will give up the accuracy, response time, and post-error slowing. And thanks to the EEG, we will look at different measures, such as error and correct response negativity, but also the error positivity, to look at the consciousness of the error processing, and other different measures. And that's what concludes my presentation. I hope I give you a good overview of our two projects. Thanks to this collaboration, and I also hope that I will have some nice results to present in a while. So thank you all.
So much stuff. I'm sure there's questions or comments or suggestions.
Yeah, just one comment. So the kids need to stay for how long in the MRI? Like more than half an hour?
The kids, they stay 45 minutes.
Is that something that they can do?
Yeah, that's okay, because we show them a movie, and often they're a bit afraid, but when they saw the movie, they're like a statue, and if they don't remember that they're in the scanner.
Okay.
And then we show them quick Tom and Jerry movie. And finally, when it is more difficult, for the resting state, we show them crust, and in this moment they move a lot, and sometimes it's clear that it's difficult and that they want to go out.
Yeah. Because I was also interested in, like, because we do more resting state fMRI. What is the parameter that you would like to extract from it? So what is the question that you're trying to answer with these techniques? Also with diffusion, but
so for the moment, I don't really know, because we're just at the beginning, and I'm not quite familiar with that. So I can't clearly respond.
Because an alternative would be to show a movie while doing resting state. So there are groups that they do that, and I could see that they, like different subjects, their network activity synchronizes with certain parts of the movie that elicit certain emotional states. And while when you do just resting state, nothing is synchronous across different subjects. So it's more difficult to assess whether you are normal or you're not, right? But maybe if you have a movie which is always the same.
Yes, it's the case.
You might be able then to have this information as a background to take some periods where all subjects should be aligned on a certain network state, and you might pick up kids that are not aligned with that and maybe investigate it.
Yes, that's right. That's the case. But we have the movie with Tom and Jerry, and that is the resting state movie. And then we have the control with the things.
But actually, with the resting state, there have been a lot of studies between preterms and controls, and there's the silence network, which is really differences between the both.
Yeah.
So there are some aspects that you can really see already when the resting state. Of course, if you go for a task, then you're just targeting some mechanisms of inhibition, as you were saying. And I was wondering, have you tried the socio-emotional abilities? Because you got the brief and the NEPSI, and I think you can even target more socio-emotional abilities on this case, because I guess you don't have many difference on the emotional and the executive functions.
Yes.
And my second question will be, these kids are doing well, I guess, in terms of IQ. So if you are preterm, but your IQ level is between the range of normal range, the executive functions, and besides the speed, I'm not sure they really change that much. I mean, I don't know. I mean, of course, we are all targeting the executive function because we know that preterm should be in the corticofrontal basal ganglia, no? But maybe emotion.
Yes, that's
the recognition of the theory of mind. An idea.
Yes, I think it would be great to investigate that. And we have some.
I do have the barretics that's sorry, you need to go through all of them again. But it may be an option.
The theory of mind, the axis that we have is via some of the auditory object discrimination, where they're action related or action free. Where there we've shown in prior work in adults that these are effective in eliciting responses from what some people call the mirror neuron system. So we'll have that in the 6-year-olds, and if it's implicitly activated, because of course it's a passive task in the neonates. The socio-emotional side, I agree, but it's interesting as well to look at the sensory aspect that's driving that socio-emotional. So I think that's some of the motivation as to why the paradigms are chosen. I don't want to speak for Marion; she can defend herself. But
why is it interesting?
Other questions, remarks, suggestions?
From the Zoom.
Maybe from the Zoom.
Cameras? I was wondering, as all the kids have with a Q, in terms of schooling, they are all part of the same schooling network, or they are in specialized schooling?
No, they're in the ordinary school, but some of them have ergotherapy, also language therapy, and some of them also have help in the classroom. But it's only, I don't remember, but maybe half of them have this either therapy or help in class. So there is still some difficulties, even if they have a good IQ and in the clinical test they are doing well, but there is some small difficulties.
Again, it comes back to the question of the emotional regulation. Is it because of an inherent emotional dysregulation, or is it, let's say, some of the sensory motor impairments that actually, I don't want to say induce, but manifest as the socio-emotional dysregulation?
That's a good question.
And then there's the other point that Marion alluded to, is the interplay between parental response and its transference onto the child. So the more that the parents exhibit post-traumatic stress disorder-like symptoms to a premature birth, it seems that can have a deleterious impact on the child. But it can also have resilience. So why do some premature kids do well and others not, even if they're all in normal range IQ? There's a lot of very open questions there. And again, it's a good thing or bad thing. I would argue it's a good thing that we have these premature kids that survive and do well, and do relatively well despite the prematurity. Now the questions emerge as to how and why and how we can reinforce that. We're getting there. And having enough kids. So the SHUV it's a big hospital, but the number of premature kids is relatively limited. So again, this is where some of the multicentric work is certainly beneficial. On the other hand, I'm sorry?
And getting the controls will be even harder.
No, the controls, it's okay. The more impressive thing that I think Marion downplayed a little bit is the longitudinal side. The fact that these kids are followed really throughout their early childhood, and that we have these exams that date from their birth, and now we're following them at age 6, 8 years old, 10 years old. I think that's where some of the causal inference can emerge. But again, this is long, slow work. I don't know if any of them have tumors where some of their tissue could be looked at, but the data is certainly there. It's a nice big data set. Hopefully you get a feel for why some of the neuropsychology and behavioral work is also important to interplay with the imaging. So yes, CIBM is all about imaging, but at the end of the day, we also want to know about people's behavior and how they do in life. Anybody in the Zoom? Sorry. And Julianne, who's here, is her actual PhD director. And I have the pleasure of collaborating with her and calling her my friend and colleague. So she's there underneath this other screen.
Sorry, I might have another comment. So these kids, you expect them to be with high IQ, so that you should not be able to distinguish them from normal kids based on this test, or you expect them to perform worse?
We expect to perform more because there is a difference between the IQ and the more either executive function or their behavioral ability. And so I think it's two different things. And we wanted not to investigate the kid who have a bad health, but we want really to see when the kid is doing well and he could attend the school. In this kid, where are the minor difficulties?
Because I mean, one thing just came to my mind. So if these kids, if they had some problems, right, with hypoxia, etc., at some point they were able to get back to the level in terms of behavioral score and everything to normal kids. It might be that the brain is actually compensating for them by using or a different strategy to obtain the same behavioral achievements, or using just more energy. And I was wondering whether there might be a way in your data to look at that as well.
Yeah, that's right. I also think that they compensate with other things, and also the fact that they've had a lot of therapy that also could drive some different strategy than a healthy kid. So yes, I think it could be really great to look at it.
Yeah. Anyone in the Zoom rooms? Any remarks, comments? No. I see we've gone over a little bit of time, and I know people are probably anxious to get their coffee.
Okay. So thank you very much to both speakers.